I'm Lisa Workzell. As I think you all know, I'm the Deputy Research Compliance Officer here at Tulane. Today we are going to be using the Box Vote electronic voting system. So I'm going to ask you all to go to live.boxvote.com and type in that PIN.
And that can be on a laptop or a smartphone.
Correct. Thank you.
Sure.
All right, what is the URL again? It's live.boxvote.com. If anybody has any trouble during today's lecture, just give us a wave and we'll try to help you sort it out.
This is designed to keep you guys awake.
Legitimate use of smartphones during lecture. All right, Dr. Prescott Deininger is our lecturer today on the topic of data acquisition, ownership, management, and sharing. He received his AB in chemistry from the University of California at Santa Cruz and his PhD from UC Davis. His graduate work concentrated on the sequence organization of the human genome. He was a postdoc at UC San Diego. Then he was a NATO fellow at the Medical Research Council in Cambridge, England. While he was there, he worked with Dr. Frederick Sanger, who invented DNA sequencing and won 2 Nobel Prizes in chemistry. While working in Cambridge, Dr. Deininger invented the procedure currently in use for shotgun sequencing of genomes. Here at Tulane, he is the Joe W. and Dorothy Brown Chair in Oncology, Professor of Epidemiology in the School of Public Health and Tropical Medicine, Director of the Tulane Cancer Center, and he also serves as the co-director of the Louisiana Cancer Research Consortium. He is a fellow in the American Association for the Advancement of Science. He has also served as a journal editor on numerous editorial boards and on many, many study sections. Last year, he served on a National Academies Committee to write a report on strengthening the disaster resilience of the academic biomedical research community. We are delighted and honored that Dr. Deininger is here today to present on data acquisition, management, and sharing. Dr. Deininger.
Thank you.
Did everyone get logged in? Okay, to Boxvote? Cool. So further in the presentation, we'll have some questions, and that will be your opportunity to put your answers in, and either digitally on your phone or verbally.
So all of that fancy introduction that I got is really just to say that I've been doing this for a long time, and I've been bit in the butt by every single thing I'm going to tell you about wrong with data. Okay, so I'm happy to keep this informal. If people have questions or anything, please bring them up. Okay, so there are several aspects. I'll let you guys read everything for yourself rather than read things themselves, but there are several aspects of why data integrity is so important. There's the broad social one that if we lose even more faith in science in our country, that's not good. It's not good for science, it's not good for the country. And every time that somebody violates the trust of science and data, it weakens us, okay? And in addition, coming down to your own level, if you violate any of the things I'm going to show and get caught, it's very bad for your own career. Even if it's not a bad violation, it can shake someone's confidence in you, your mentor, your place you're trying to get a job, or somebody who's reviewing your grants. And so trust in somebody's ability to handle their data and so on is absolutely critical to what we all do. So I'm going to talk about acquisition of data, talk about objectivity in that acquisition. We'll talk a little bit about the laboratory notebook and the whole spectrum of notebooks there are. We'll talk about some of the errors and ways that data can sometimes be misused either by accident or through misunderstanding or through inappropriate behaviors. And then we'll talk about data ownership. Okay, so acquisition. So acquisition is basically do your experiment, you accumulate data. But proper data acquisition requires record keeping. There has to be a solid record of whatever you've done. And you'll need to store that data so you can analyze it later. You need to store the data with good record keeping so that it's not tainted. And what tainted means is that if you've taken your notes on a back of a towel and tucked it in your pocket when you're done with the experiment, well, if you pull that out a year later, it's not going to make a very strong case when you try to write the paper that you really did the experiment or that you should get a patent for that huge money-making idea you had. You absolutely have to have a good notebook to get a patent and to establish when you made your discovery. And you really shouldn't depend on your memory. And this is probably the one that probably most people get bitten by. You're all doing the same experiment day after day after day, and you say, "Oh, I always know what this experiment's like," and so you sort of skip some of the details, and you don't say, and then you gradually evolve how you're doing that experiment, but you don't bother putting that down because you're doing it every day, and so on. And it turns out 3 months, 6 months later, I've learned the hard way, you don't quite remember exactly what you changed or what you did and so on. And so it's very important that your notes be solid so that you can look at it a year, 2 years later, because sometimes it takes a while to get your publications out, and you can remind yourself what you have. Objectivity. So in terms of data, you got your raw data, something you might be writing in your notebook, like cell counts, or you might put some data in out of a machine that has NMR peaks or something like that. You also have process data, which is also important and should be part of your notebook process. So you see the evolution of how you're developing your ideas in your notebook. So you're going to be putting charts and graphs and trying to assemble and analyze your data in different ways. You'll also have various things like you'll need to take notes. So you're doing, in this case, if you're doing a mouse study, you might want to just note some things down because sometimes you don't know where something will take you. You have an idea that you're testing, a hypothesis, but maybe something else will happen. And if you see something weird, sometimes the weird things you see turn out to be your next project. Okay, so making a few notes, and then there's also instrument outputs. Some of these I consider real data, pictures of gels and so on and so forth. But some of them are things you're going to develop that are useful for presenting or analyzing your data. And then the last data would be, let's say you develop an animal model and you study it for a while. That animal model is important and needs to be preserved so that somebody who comes up with another idea, or you who want to study some other aspect of it, can take advantage of all the past studies you've done on that animal, and then somebody else can build on that. And the same is true there's all kinds of things you should be storing away from your experiments that you developed that other people might want to use to follow up your work. Okay, so all of that's critical, and all requires a lot of thought, it turns out, to do properly. Now, objectivity, in general, hopefully that's obvious that we have to be objective. We're scientists. And so that just means you're not biased. And one of the biggest biases is personal gain. And this can be subtle. It doesn't have to be, I'm going to make $1 million. We all see in the news times when people violate things and make a ton on a medical thing that turns out they had a conflict of interest on or something like that. But we all have little bits of personal gain to be worried about. Getting your paper out, getting the next grant, pleasing your mentor, all of those things can be personal gain. And when you're doing an experiment, they can bias that you want to come up with data that looks, that will please those particular groups. And here, this is another thing that I still run into as a reviewer on grants and things. The one thing that will absolutely kill a grant for me, and to some extent papers, is if somebody comes up with a hypothesis, it seems like a good hypothesis, but as you read the grant, all they're trying to do is prove the hypothesis, as opposed to testing the hypothesis. Okay, all they do is design experiments that would show you things that would support the hypothesis, but they never come up with the experiments that might come up with an alternative to the hypothesis. And it might be that because once you try all the things that are supposed to test the hypothesis in a grant, if that hypothesis isn't true, then you have to have some ideas about what you're going to do next. And if you don't tell me what you're going to do next, it makes it feel like you have the idea that, this is my hypothesis, I'm going to prove it, okay? As opposed to, this is my hypothesis, I'm going to test it. And you have to look like an unbiased scientist. You have to be an unbiased scientist as much as you can, so that you can change your ideas and take full responsibility for whatever you do. Okay, so here's some of the personal gain things. Desire to please one's supervisor or mentors. Desire for promotions and advancements. Young faculty are under a lot of pressure, to get tenure and be advanced and so on, and really want high impact papers and so on. Improve your chances for grant funding. Again, you want big ideas, get publications. But the important thing is there's data, and you can't change them. You shouldn't do anything that manipulates them inappropriately. There are some acceptable types of data manipulation, but you don't want to be picking and choosing and saying, well, this experiment proves my hypothesis, and there must be something wrong with that experiment because it didn't. It was against my hypothesis. Well, there's only very limited examples where you can throw out that one experiment and keep this one. You really have to consider both, unless you know that there was some horrible flaw in that other experiment. Okay, so we're going to talk about a couple of case studies. So a PI outlines a theory to a student who is involved in acquiring data to confirm or refute the theory. The PI explains their anticipation of finding experimental data having certain values. The grad student generates experimental data and gets approximately the anticipated values. The data are published. The PI gets great recognition. A grant is renewed, and the graduate student gets a PhD. Everybody's happy, right? It all worked out. The problem comes, and why is this not turning off? That's not going to be good for my. The problem comes that the next student coming in working on the project can't reproduce the data. And after they look around for a while, they actually find that the first graduate student chose only the data that supported the hypothesis, and whatever is ambiguous, they left that data out. Okay, so that probably it wasn't true. Now, I can think of multiple cases where a graduate student comes in to work on somebody's grant and just accept that the previous project was done properly and move on to the next thing. So one thing I've learned is that as a grad student, even though you're depending on your mentor to train you and so on, you also have to understand the background of your own project, look at the past data, convince yourself that you're working on a project that's solid. Because ultimately, even if it's a PI's mistake that you move on, ultimately, who gets hurt most if your project turns out not to be valid, right? It's a student who spends critical years and then doesn't move forward. So it's really important not to just take everything on faith, but to really understand your project and the background behind it. Okay, so I think this is where we get to the Vox voting. Yeah. Should the PI have not mentioned her anticipated results, his or her anticipated results? We have to do these one at a time, don't we?
Yeah, so just type in your feedback or also raise your hand and speak your feedback.
Real time?
Yeah,
Or people not voting?
hit refresh. There you go.
There we go. Okay. So that's a pretty strong yes. We'll move on to the next question. Should the PI have more closely supervised acquisition of the data to verify their accuracy? Okay. And should the PI have insisted on a more thorough check of data reproducibility? Okay. Is this one that could be answered? That's
Yeah,
okay.
it creates a word cloud.
Okay, a word
Type in
cloud.
free
We're gonna have a
text.
word cloud. What should the PI do now about the situation? You guys can probably text better than I.
It's not popping up.
Oh, no. That's my.
That gave you time to think about your answer.
Mercy.
Retraction, I see that. Mercy, yeah. Retire. Please, yes, yes. Okay.
Still, probably some more.
Correct. Everything. Okay. Not sure why they're moving around, but they might be consolidating in there. Retract is getting bigger. Oh, I thought it said meditation is mediation. You gotta meditate when you're in stressful situations as well. Well, this is actually kind of fun. I enjoy this.
Cry.
Cry. That's a good one. All of those are gonna happen. But let's talk about this. Okay. So the PI's job is to supervise the design of the experiments and processes required. Okay. So there is a negligence on their part to some of those things. Whether the PI should, I don't think it's absolutely clear on that first question whether the PI shouldn't have said what they're expecting to see. Where they might have led it astray is they shouldn't make it that this is the only thing I might expect to see. It's worth talking about the experiment and say, hey, look, I think the most likely result is this. But if we see this, that means something different. Okay, and that's actually how I write my grants when I write them. I walk the reviewer through my thought processes of, if I get this result, it means this. If I get that result, it means that. And if I get that result, it means I have to do this other experiment. It takes me in a whole different direction. That's what a PI should be doing for you. It's okay for them to be excited about a particular direction it's going, but they have to make sure that you're thinking about the other possibilities. They absolutely have to be, when I was a young faculty member doing just DNA sequencing, when I taught somebody how to DNA sequence, I would sit down, and after they'd read all their sequencing gels, because we had to do it manually in those days, I'd sit down and I'd read their gels myself. It can be very laborious to be a PI overseeing a lot of these data. As you get into a bigger and bigger lab, the type of oversight gets more difficult because they can't go through every piece of raw data. But there ought to be some structure within the lab that looks at your raw data and helps you assess it. And I'll show you some examples of some stuff later. scientists do have this duty to avoid contaminating the literature with incorrect information. And so basically they should either decide we've already disproven this previous paper, or at least redo the experiments independently. And if they can't reproduce them, retract the paper, or at least print a correction for the paper. And maybe the whole paper isn't wrong, maybe only a certain part of it. You got to deal with that effectively. Okay. Yeah. Another case study. A PI acquires data that shows a wonderful correlation between administration of a drug and a response. About 10% of the data, however, lie far removed from the predicted values. There are explanations as to why these data are different. For example, some experimental parameters have not been well controlled and were different from the experiments in which the results deviated. The PI chooses to ignore the outlying data in the publication. Is this okay? Is it okay to ignore outlying data with an explanation? Or should you have to show the outlying data that might put questions in people's mind about your hypothesis or your findings? Okay. We're leaning towards the high ethics side, okay? And again, it's not clear. The devil's in the details on this one. The question here would be, why are you throwing those data out? And the best way to do that is to have some criteria ahead of time about what might happen, why you might throw data out. For instance, is this if your mice get loose and go running around and all of them get mislabeled, okay, or potentially mislabeled, or get exposed to other things, you should probably can those before you even look at the data and go back and start a fresh batch. That would be even better if it's a little bit shaky to look at the data hoping that it will fit and then say, I'm going to throw it away later. If you have an experiment that goes a bit awry, it might be better to just start it over again. But these are some of the questions you have to wrestle with a little bit, as you see, because there are always a few difficult to control things in an experiment. But to throw data out, there has to be something very striking later on with a particular experiment. And you're always a little bit safer ethically with bringing things forward. Now, it would be hard to explain in a paper that this group of mice got completely exposed to something different. That's actually just detracting people from your experiment, because in a way it was a different experiment. So I can see why you would leave them out sometimes, but it would depend on exactly how big the perturbation of the experiment was.
Dr. Deininger.
Yeah.
Is it common for an author to request a correction? So like in the first example, if new information is found later, I mean, is this something that researchers should expect will happen at some point in their career?
I've seen a reasonable number. It's not super common, but I've seen a reasonable number of corrections where an investigator says, "Oh, I've learned this and that." And a correction, of course, doesn't remove your publication from the publication record or anything like that. Now, perhaps even more common is a complete withdrawal. I've found that both journals and authors resist withdrawing papers because, for one, it makes them look really bad, and it also takes away something from their CV functionally. So both of those things happen at times, not terribly frequently. Yeah.
I have a question about the outlying data. I wonder if it varies by discipline. I've seen, for sure, in social science articles that are published where people talk about data correction in terms of normal distributions and outlying data. And so they are omitting data. But
there are some statistical, valid statistical approaches, more than two 2 standard deviations from the mean, sometimes you can throw data out. And of course, you need to think about what it is. Sometimes, for instance, if you're dealing with mice or people, there can be, with people, for instance, you've got crazy genetic variables in there that could make a few people look really different, okay? And you could see that. So I think how you would do that would depend on what the thing is. But yes, there are statistical valid approaches in many experiments to leaving out outliers.
And does that have to be explained in the manuscript at all, or is that something that's
It's
standard?
always safer, and I'll be talking about some figures and some adjustments of data or whatever, but it's always safer to include that, even if it's only in the supplemental methods or whatever, to tell what you did. Yeah.
A slight deviation from the question around outliers, but around data and recoding data and things of that sort. What about procedures around missing data? So like imputing missing values and things like that.
Well, you should clearly state what there are some great methods in genomics, for instance, haplotypes and HapMap kind of stuff for putting things together. But you really should be explaining what you're doing there. And ideally, you'd be taking into account any of the into the statistical analysis of your data as well, which gets probably more complicated in statistics than I can handle. But yeah, there's clearly times when it's, especially when you deal with humans, but in studies where you have missing data and you have to figure out, best with the help of a statistician, whether there's an appropriate way to analyze those data. Something I'll tell you is that I learned the hard way many a time is if you're doing a study that you think is going to need statistics, talk to the statistician before you do the experiment, because they can come up with some really clever experimental designs to protect you from some of these issues. Okay, notebooks. I've actually found in some labs, people just sort of expect students to take a notebook. There's not training in notebook taking. So it's important that you learn from somebody who makes a good notebook how a notebook should be. It has to be bound and numbered to be valid for patents and so on. You have to have a date wherever you're working with. I never liked the approach of having to date and do, like if I were doing 3 or 4 completely independent experiments, they'd all be on the same page. But that's really how it has to be. If you want, you can get different colored pens and do your different experiments in different colors. You got to figure out how you can organize it. But it has to be dated with each experiment in a linear fashion so that you have a timeline of all the things you've been doing. This is something a lot of people don't do. A lot of people just put the data, just the facts, okay? But I like to put why an experiment is done. I like to put some of my thinking on the page. It doesn't have to be more than a line or two. But that, for one, if somebody else is ever trying to read your notebook, heaven forbid, it gives them a little guidance in looking at it. But for yourself, a year later, you might have totally forgotten why you did a new variant on an experiment. So put why. You obviously have to describe what you did, okay? You should, as much as possible, put your raw data in there or perhaps the location of it. And this is, like I do a lot of next-gen sequencing now. It's just billions and billions of bases of sequence and huge files. So there's no way I can put all that sequence in a notebook. The best thing I can do is submit it to a database where I can then link an electronic link to that database. But until that, I need to sort of tell myself what's the name of the file, what hard drive is it on, and so on. But we have the same thing with all these electronic images. We don't always print every single one out. Plus, sometimes you need the electronic image for proper quantitation later on. So you need to know where the files are. So you really ought to be linking your computer to your notebook in some way so you can find your data and have everything organized. Here's this experiment, here's that experiment, have all the data put together as much as you can. It's up to you with your particular type of science, figure out what your best storage approach will be. And it should be backed up if it's on a computer. And then you should actually put a few words, just like you put a why, What did you learn from that experiment? What are your conclusions? Okay. What problems did you see? What are you going to do next? Has this experiment, did you see something that was not quite right? Or did you say, oh, the statistics didn't quite come out, I better do this 3 more times and see if I can get something that's statistically found? Whatever you do, just a couple lines on here. And we have this here. I've tried to get people in my own lab start exploring electronic notebooks, because a lot of the problems that I just described could be fixed in an electronic notebook where they can all be linked, and some of them even have all kinds of neat data things. On the other hand, I think there's a learning curve to getting into electronic notebooks, and none of my people yet have been willing. Do you have many have you run into many examples at Tulane of people using an e-notebook? Some of them are free.
Well, how about
different strategies?
Are any of you using
Yeah.
electronic?
Okay, we got a couple.
Cool.
And do you think it's easy?
Yeah, it's like he uses a website called Lab Guru, and it goes back like 2011. You know what I mean? So I can go back, like I'm repeating something, and so I can go back to 2011 and be like, well, like what did they do and why did it go wrong? It's like,
And a good thing is with an electronic notebook is, in theory, it's backed up. You don't have to worry about a fire burning it or whatever. Of course, you're also counting on them really backing it up and not screwing up or not going bankrupt and losing everything. But at the very least, you could probably print it all out if you really had to or whatever. So e-notebooks, I think, have got to become more prevalent, and they would solve a lot of the problems with all these big electronic data sets we get these days. But I haven't seen that much use of it. And it actually would be great for somebody who's an expert to give a nice presentation to everybody else about how they use their e-notebooks.
Anybody else using?
But like one thing is that with e-notebooks, it's easier for a person to edit or change things.
Well, I haven't experienced that. In theory, that should not be true. In theory, once you've put something in, I mean, for instance, in a notebook, you can write it, you can change things in it, but you change them by drawing a line through it, leaving that record of what you said before, and then writing what you think. So that, if you made a mistake, you can fix it. You should be able to fix it, but you shouldn't be able to erase anything you ever put into a notebook. And hopefully that's still there. Any notebook that lets you go back and completely edit it without a timestamp on that new edit is not a good notebook, in my mind.
Have you experienced that though, that you're able to change it, or someone was able to change things?
Yeah, it's like just like one of my previous PIs like would criticize me, saying like she required me to both have an electronic and a print version. So that's what I was saying.
I think in most of the ones I did look a little bit. Most of the ones I looked like they had some reasonable timeline on it, and even though you could probably go back and make notes, the notes would be redated, and so it would be crazy not to have that. I think in one. So I wouldn't use one e-notebook that wasn't firm in terms of what you put in, never getting it completely eliminated. We'll talk about electronic images too in a minute. Okay, so a graduate student begins a project. This student talks about the project with his advisor. Reads up on it. The advisor never mentions notebooks, assuming he's just going to follow somebody else in the lab and how they make a notebook or already knows how to do it. And the guy begins the work and gets some great results. They discuss it periodically. They're getting excited. They look at the films. So they're doing all kinds of stuff. And after a while, 6 months is pretty quick. They write up a raw draft with the data and so on. The advisor has questions about the raw data and asks to see the notebook. There is no notebook. There's a bunch of pieces of paper stuck in a drawer that the student wrote little things down on, but basically there's just a bunch of gels and so on and so forth. Most of the things are undated. And he can't even find all the data that went into those figures that were there. Okay. That brings up a question. What should be done? Box. Do we have that? Okay. We got the timer running. Remember, this isn't published yet. They're really preparing a publication on an exciting result. Redo, repeat. Rerun. Start a notebook. That's a good one. Award. Plain. Okay, I think we're keeping some fairly consistent pictures here, where with repeat and redo together being 90% of it, or a big chunk. Okay. And who? Yes.
Well, so is that what you would advise?
We'll cover that in a minute, but yes, in actuality, that data, for all intents and purposes, does not exist. If it's not in a notebook in a way that can be backed up, first of all, you shouldn't trust it because you're trusting a memory that this goes with this and so on and so forth. But second of all, if an editor wants to see your data in any way, if there's ever an ethics review on your paper or anything like that, and you try to show them a hodgepodge of little strips of paper, that first thing they're going to do is think you're an idiot, and the second thing they're going to do is make you retract the paper. So this project, for all intents and purposes, was a pilot project. It gave you some idea that there might be something exciting to be done there, but you have to do each and every experiment over again. Okay? Who's responsible? PI, the student, your mother.
Administration.
Yeah, the responsible conduct of research folks. Research compliance, yeah. Advertising. Not really. PI is dominating, but student is up there quite a bit. The committee, the committee has some responsibility. Okay, I think we're coming up with all the same words in proportion. And basically, I do think, so the data don't exist. So that was the first question. Second one, it is the advisor's responsibility to train you. And the student should not be expected to know what they don't know. Okay, so if they don't know about a notebook, somebody has to point it out. Now that's something that, on the other hand, the student should notice. And the other side of it is, who gets hurt by this? It's a student. So the end result is, yeah, the PI is responsible, but most of the pain is felt by the student. So that's why it's important that you guys keep your eyes open. You guys evaluate what you're doing all the time, and don't just mindlessly do what your PI thinks you should be doing, okay? It's way more fun to be critical, to think through things, and think about whether you're doing the right thing all the time. Okay. Now here's a pretty good notebook. This is because it's not easy to read. Basically, this day they must have done a Western blot, and they're talking about the secondary antibody. You notice here's some data, and they've labeled each lane. And over here is an example of how they'd set up such an experiment. They actually have a template for that experiment that they use all the time, and so they put in how much is transfected here and what goes into each cell type. And somewhere they probably tell what kind of cell they're using and transfection method they're using. And I think somewhere in here they have a few things, just a line or two about what they should do next with those things. And the handwriting is manageable. It's better when I'm looking at it on my own computer screen. I can actually read most of this. Okay, here is another example of a notebook. Okay. I've stared at this forever. And
There are true lab, these are real lab notebooks.
Yeah.
We didn't make them up.
I've stared at this forever, and I can't figure out anything. I think, these are some experiments that were eventually put on a gel and analyzed somehow. And who knows, there's some sort of a matrix here. This is more of the same thing. And this is more from the same notebook. Some washes involved with something, so on. And then here's some data. Notice it's not labeled. I don't know which of these corresponds to which of these. Okay, bad notebook. Not only could a stranger looking at it, not even a knowledgeable stranger in the field, not figure out what went on for sure, but I'm betting that 6 months after they did that, whoever wrote this notebook couldn't describe to me exactly what they did for every experiment on that thing. Okay, so work at it. Yes.
I noticed on the good lab example, there was something printed from a computer. And for folks who maybe are more quick at typing, but they're still in the lab where written lab notebooks are used, this is something that could be done, that you could type your notes and then print it and stick it in the book.
It's a good question. Formally, of course, the point of bound pages is as though things can't be removed. Okay, like if you just have a binder, you could remove a page, rewrite it, put another page in. It would do the same thing as editing your e-journal. You don't want to have that. So again, this is actually an experimental description. The act of taping it in would probably make a mark when you tried to remove it. So taping something like this in, but I don't know about taking a whole separate page and just flipping it in. Maybe typing something out and taping it in would be okay. I'm not quite sure.
Well, I've read once that the best practices for something that is going to be taped into the lab notebook would actually be the date and sign, where you're writing on both the tape and the paper.
Yeah, see, but something like that would actually validate exactly what you're doing. Of course, if you're that meticulous about the typing and stuff, you probably ought to be thinking about e-notebooks, because they'll take care of that for you right away. But yes, that sounds great. You need some way to make sure that if somebody's trying to change anything, it shows. I've heard of examples of where the FBI brings in handwriting analysts to, like with patents, to prove whether somebody made a discovery on this day or that day, and they do a handwriting analysis to make sure the same person wrote it in about the same time, and that the ink is from that period or something like that. Who knows? So it can get pretty forensic. See, most of us won't have to face that, but I'll tell you, making a good notebook, it's just a no-brainer. It's such a pain to redo an experiment where you already know the result and you've already spent money on it. Your PI is going to be pissed off if he has to spend money on that experiment again. So that's important. Okay, scientific errors. That's the idea that you do something that just pushes things a little bit your way. There's another step up where at the experiment, you sort of ignore data you don't like. You say, well, that experiment couldn't be right. It didn't prove my hypothesis. There's some things you can do that are inappropriate at the analysis level, like forcing funny groups together or using an inappropriate statistical treatment. Again, there's all kinds of things in interpretation level, personal bias. If you come up with an unreal model that you're testing, then your data won't be very solid. I just reviewed a paper on that where somebody was talking about gene expression, but had a completely unreal model about where the background would come from. Ultimately, fraud, the big devil there, requires an intent to deceive. So we go everywhere from slight bias to incompetence to cheater, okay? And you have to have the intent to deceive for it to be called fraud, but you can look like a pretty bad scientist with any of these other things if they're looked at. So sometimes your sample error, you can have a sample size that's too small, or you have a selection bias in your sampling where you ask right outside the Democratic Party what whether they'd vote for Trump or something like that. There's all kinds of things you could do. You have to look for ways to create an unbiased approach to that. In my world, where for instance, the experiment I was first exposed to this was we were measuring cell growth rates by incorporating radioactivity into the nuclei. And when you do that, you get on a scope and you're reading silver grains over a nucleus. And there's nuclei with lots of silver grains, and there's nuclei with no silver grains, and then there's some with just a scattering of silver grains. And there's actually a judgment call at what point you call it a positive and which point you don't. And if you know what you're looking for, you could maybe use a little different positive on your controls than you do on your experiment, okay? And the way you get around that is double blinding the experiment, not knowing who your controls are or versus the experiment. Or even what we used to do is have several people in the laboratory read the same things, count them up, and they don't know anything, and see if there's an agreement. So you have to figure out ways to pull out your personal bias whenever there's judgment calls going on your data. There's always screw-ups, measurement errors, machines not calibrated. I can't tell you how many times I've seen people screw up DNA concentrations for whatever reason, because there's particles floating around and they didn't spin the sample before, put in the spec, and they got light scattering instead of absorption. And then you put it on a gel and suddenly there's way less DNA than they said they were. So you have to double check yourself as much as you can. Make sure your machines are right and keep your eyes open for things that go wrong. Things can be misclassified. And there's all kinds of other errors. Things like things going wrong with an incubator. It might be, hell, in my building, it might be the power going out for 3 hours sometime, and CO2 going off in the incubator. You may not even know it initially, okay? Or it could be, humidity changes and gear phase of the moon. You have to keep your eye open for anything that might be changing your data. And then there's fraud. There's all kinds of things where you can screw up, but fraud requires the idea that you deliberately intended to deceive. And all of these has happened in science. People who never ran the experiment and made up their data, people who just didn't repeat an experiment but faked the repeats, fudging data, deliberately ignoring data that doesn't fit your model, and misinterpretation. So, I mean, this is bad news. This happens in most worlds. If it happens, you're actually not allowed to write grants to the NIH for a period of time. It would probably be a basis for removing tenure from a faculty member. It could, there are some serious potential consequences So I hope none of you are ever even think about something like that. This is too small, so image processing. I actually gave you guys a link somewhere to some stuff about image processing. The most important things is images are data, and the original image is your data. You should actually always keep a record of that original image, even if you've had to do something to remove background later or cropped it or whatever. Keep the original image as your data file, okay? And that should be saved. And any manipulations you do should be relatively simple. They should be things you can explain in a paper that nobody minds, which would be lightening or darkening a whole image. That's pretty well considered. And or removing background if you explain how you removed background by using, say, a section of a gel that didn't have any sample in it as the background. It's even okay to cut out parts of the gel, to crop them, okay? What's not okay is to crop different parts from different gels, put them together and pretend that they were one gel at any given time. If you're gonna put together things from different gels, you gotta like leave lines around them and show that they're different gels and show why you can put them together. There has to be some common size standard or something that allows you to put them together. So you're allowed usually to manipulate the whole image as long, although not with things that aren't sort of linear, and you have to use some common sense here. Basically, you don't want to. You need to make sure that what you're comparing is real. So well, let's see. I don't think I have an example of that. One example though would be when you do fluorescence microscopy. How long you expose changes the intensity. Okay. So if your controls are exposed for 10 seconds and your experiments are exposed for 30 seconds, your experiments look lots brighter, okay? But that's not a fair comparison. They have to be done under identical sets of conditions. If you're going to change the intensity, you do the controls at 10 seconds, the experiments at 10 seconds, and then the controls at 30 seconds, the experiment at 30 seconds, so that people can see both sets of comparisons of what you've done. So that's mostly what you want to be able to do. You never go down and mess with at the pixel level. You always want to keep your magnifications consistent. So all of that's in the slide. Okay. So this is a site that has a couple interesting things with figures. It's an NIH site. I'll let you guys look at it when you want. There's some really... There's examples of things that people found in the literature where people had faked their data, turning images around and putting them as a different panel and so on. And they were caught by people just reading those papers. Okay, so, the bad guys get caught. And so they have real world fraud examples and some general purpose descriptions of what's valid to be done on those images. So let's look at a couple of images. These are some real images that I analyzed once, and this is what was submitted to the paper, to the journal, and you've probably seen this kind of thing where people do different treatments and glue these just the part of the Western blot that compares. Now the problem with this is, you get up here, and they're saying there's no signal here. Well, when you look at the raw data, and this is where PI should take a look at the raw data and say, that doesn't look quite right to me anyway. Okay, but if a PI were to look at this and they'd done any of these experiments themselves, they'd say, well, you know what? This looks like there's been some air bubbles between the membrane and the gel that screwed up the transfer and weaken the signal. And so I can't trust what's here. You can even see a little bit of a band right here that this bubble has gotten rid of. So if you're used to looking at data, and a PI should be used to looking at data, and the PI, a student might not catch this, but a PI should have caught this. In addition, what should the student have done that would have caught this? you should have repeated the experiment multiple times so you could get, real reliability and maybe statistical analysis on it. And if you do it multiple times, the odds of the air bubbles being in the same place every time are pretty slim. So that's why repetition is so important. If I have a really big discovery in my lab, it's really good, and we're saying, oh, nature, here we come. I almost always have somebody else independently in my lab attest to that. Here's another one where you can see a bunch of original data. This is how it came together. There's a lot of creepy stuff here. One is, you notice that the gels, it seems like each set of lanes was a different set of gels. The whole point of controls is an experiment should have one variable and only one variable. And when you run the samples on different gels, suddenly you've increased the number of variables because you've got loading errors and transfer errors and all kinds of other things between the gels. So ideally, you would have run one gel and then sequentially probed it with each antibody. Sometimes you can't do that. Sometimes you have to bite the bullet and run multiple gels. But this one was creepy because I'm not even sure there's a lane there, and that was meant to be. That's this spot over here. I can't see evidence without a control to show something's in that lane. I'm uncomfortable. In addition, you notice there's different numbers of lanes in different samples. They're not laid out in the same way. That's just not right. That almost looks like somebody taking different experiments and trying to use one experiment as a control on another experiment. That's just not right.
And Dr. Deininger, the figure on the right is what
this was in a publication.
Right, that was in the publication. It was questioned, and when we asked the PI for the original data, this is what we got. So as you can see, they came from multiple different experiments, not one, as Dr. Deininger said, should be.
Yeah, so very bad practice. And so this is all just, most of this is common sense if you stop and think about it. I'll just mention briefly, because we're running out of time, ownership of the data. First, the good news is it used to be that you had to get NIH's approval if NIH funded your thing to develop it. Now the Bayh-Dole Act gives it to the university, basically. Tulane turns out to be very, very nice. Tulane keeps about half of the money and gives about half the money to the investigator who generated, even a little bit more back to the laboratory as well. So this has helped generate, get your data out into practice because the university basically owns the data. And that's the most important thing here. I'm trying to see, do I have that? Yeah, okay, I do have it. Here's some web pages, so you guys have access to my slides, I think, that talk about things that there's all kinds of information on these Tulane web pages related to keeping data relative to these things. For instance, you have a whole different set of rules about keeping human data, and HIPAA compliance and so on that you guys should all be taking lessons from, at least if you're going to do human data. And Tulane owns the data. This is something a lot of people don't get. You don't ever get to walk away from Tulane with your notebook. In fact, it should really never leave the premises. You can copy your notebook and take a copy. That's what I've always done when I left the laboratory. I copied my entire notebook, billed it to the previous lab, but had copies then of everything I had done that were useful to me as I moved forward. All the raw data belongs to Tulane. You can, again, copy some of that, although only if your PI is really going to let you. This should be talked over with a PI. It's the PI who manages the data for Tulane. In theory, the PI shouldn't even take away the notebooks if they move to another place, because it would be Tulane that would have to defend that work that was done at Tulane if there were ever a problem. Okay, that's it. So you guys are all responsible data managers now. Anyway, I'm available if you ever have questions about any of this stuff. Thanks.
So if anyone wants to stick around and ask some questions, feel free. And make sure you sign the roll sheet and please leave us your name tags. Way out.
So I just had a quick question
Yeah.
about case one. So I worked with a PI in which she had to actually fire the staff member.